Hi to everyone who has joined so far. I'm going to give others just a couple extra minutes to join us, so we will begin shortly. Hello, everyone, and welcome to our webinar, Expanding Neoantigen Discovery with COSMIC. I'm Caitlin McCann-Jankovitz, and I'll be your moderator for today's interactive event presented by LabRoots and brought to you by QIAGEN Digital Insights, or QDI. To learn more, please visit digitalinsights.qiagen.com. In this session, we want everyone to be a part of the action, so we ask that you please take part in the polls we'll be sharing throughout the presentation. Our speakers will use your feedback during the presentation as well as after to improve future trainings and resources, so your participation ensures that everyone gets the most out of this experience. If you miss any poll questions, run out of time to answer one, or simply wish to answer all of the questions in your own time, you can also take the survey located on the right side of your screen. This survey includes all the same questions we'll be asking as polls, so feel free to participate in one or both options. We highly appreciate any and all of it. Another way we encourage you to participate is through Q&A. To do so, simply type your question into the Q&A box located on the far left of your screen and click send. We're going to be taking several breaks throughout the webinar to answer questions, not just at the end, and we will address as many of your questions as possible. If we run out of time, don't worry, we'll follow up via the email you provided at registration. One more thing to note, this webinar will be available on demand following the live presentation. LabRoots will alert you via email when the recording is available for replay. And with that, it is time for our first few poll questions. So, question 1, what best describes your work? Academic or nonprofit research, clinical sample analysis and reporting, commercial drug development, or commercial biotech? I'll give you about 10 seconds to answer. Okay, next poll question. How do you typically use COSMIC? In this one, please select all that apply. As a web portal to learn about individual variants, as a data source to integrate into your pipelines, for classifying mutations based on pathogenicity, to link variants to cancer types and available drugs, for cell line selection, and I don't currently use COSMIC. All right, and final question for right now. Please identify your most common query types or use of COSMIC data. Again, please select all that apply. Gene-centric queries, disease-centric queries via the COSMIC Cancer Browser, large-scale bioinformatics via COSMIC download files, validation of pathogenicity of a variant or driver status of a gene, mutation signatures, and clinical relevance, including actionability and drug resistance. Again, I'll give you a few seconds to answer. All right, and with that, I would like to now welcome our speakers. Dr. Kyle Nilson of QIAGEN Digital Insights, our primary speaker in today's event, as well as two members of the COSMIC team at Wellcome Sanger Institute who will be joining us for Q&A, Dr. Jennifer Wilding, Scientist Curator, and Rebecca Steele, Clinical Curator for Actionability. For their full bios, please visit the Biography tab at the top of your screen. And just one more thing before we get started, our legal disclaimers. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, please visit www.qiagen.com. A license for Cosmic is required for commercial use of data obtained from either the website or download files, which ensures the continued growth and maintenance of the resource for academics and industry alike. For additional details regarding commercial use of Cosmic, please visit the URL you see here or contact bioinformatics sales at QIAGEN.com for complete terms and conditions. And with that, Dr. Nilson, I'll pass it to you to get us started.
Fantastic. Thank you so much for the introduction. And everyone, thank you for taking the time to attend today's webinar on how we can potentially use Cosmic to improve characterization of peptides and identification of potential neoantigens for research. So in addition to using the Q&A box, we love questions. We love finding out about use cases and tackling them. We also want to be sure that you are aware that you're always welcome to contact us either here at QIAGEN or the Cosmic people at Sanger. So on screen are two very handy email addresses. First is the tech support for our QIAGEN Bioinformatics. And then second is also the Cosmic email address for their help desk. So if you ever have trouble figuring out where are specific fields in the files, say you want to link several tables together, shoot us an email. We'd be happy to help sort that out. Likewise, if you want to know more about curation, you want to know more about how you could potentially use Cosmic in your research, again, we'd be happy to discuss. So for today's presentation, I like giving a bit of a taste up front of essentially the kinds of things that we're going to show and discuss today. For starters, I couldn't start this presentation without mentioning what Cosmic is. And in this case, it's just simply put the gold standard for somatic mutation variant information. So if you are a scientist and you are interested in looking for the most comprehensive or the broadest coverage of any kind of cancer variation, Cosmic is likely at the top of your list of places to go or resources to use. And with that comes from consistent curation, that comes from really taking advantage of careful, controlled metadata terms, and of course, the decades of consistent output that the Cosmic team has put into this resource. So all of these things mean that you can then potentially implement this Cosmic data into your own research pipelines. So Cosmic files are available to download both for academic and commercial users. And today we're looking at an example where one particular lab really needed to bolster the peptide libraries that they could align against. So for today's use case, we're looking at a really cool paper where the authors in this case took advantage of Cosmic in a rather unique way that turns out to be worth sharing. In this case, what they wanted to do is they used Cosmic. They took advantage of the curated metadata in terms of where the samples are, what kind of cancers they have, and they were able to create a tissue and disease-specific library of potential peptides that they used for their proteogenomic analyses of immunopeptides. And ultimately, what they found was that by using this approach, as opposed to the typical standard, which would be taking advantage of, say, whole exome sequencing for whatever material is available for a cell line, they were able to improve detection of potential peptides of interest. And of those, they were able to identify more neoantigens. So this was something that allowed them to really take advantage of their data. They started by using HepG2 cells just essentially as a proof of concept, but the idea would be we can go ahead and take advantage of this kind of method to start dealing with other sample types and possibly move this into clinical use. So overall today, while talking about all of those particular topics, I want to first start by just giving a quick introduction to what expert curated content is and the kinds of resources that we at QIAGEN like making available, as well as details about COSMIC, which is one of the best, again, somatic mutation resources, and we're really glad to partner with the Sanger team. After that, we'll go ahead and focus on the use case, looking at that expert metadata, in order to take advantage of and focus just on specific cancer types or just on specific tissues. We will go ahead and I'll share my screen. I'll show how you could potentially approach that. And then finally, we'll go ahead and briefly discuss some of the results from the manuscript. But first, we want to go ahead and give that quick introduction. So what do I mean by expert curated knowledge? Here at QIAGEN Digital Insights, something that makes me extremely happy as a scientist is we really focus on the best quality data. And we take a lot of time to clean that data up to make sure that it's searchable, to make sure that it's valid and mistake-free. And we have quite a broad variety of data and information that we make available. So we're not necessarily talking about our curated research findings or real-world omics data today. Those are two extremely useful data types. The curated research findings are where our team of scientists will read manuscripts and identify specific nodes, in this case, proteins, drugs, diseases, adverse events, and they will identify what the relationships are between those nodes, and they will characterize those in a directional manner. Does this protein lead to an increase of phosphorylation of the second protein? Or is this protein correlated with increase of a disease activity? These essentially form a knowledge graph that we make available through our biomedical knowledge base. The H.D. stands for human derived. And that ultimately is one of the biggest sources powering Ingenuity pathway analysis, which is a really nice tool that's easy to use for scientists. In addition, here at QIAGEN, we also will curate omics data where we will download studies from GEO, SRA, EBI. We will take the time to check all of the metadata and make sure that there aren't any mistakes. I know when I uploaded my own research to GEO, I would just drag and drop and get K562, K563, K564. Those metadata mistakes, as well as swap samples, inconsistencies between manuscripts using the same data source, all of those get checked, and ultimately the files get realigned and normalized to make it possible to then search and cross-compare all sorts of expression data and proteomics and other sources of information. So those are available in OmicsSoft Studio as well as our APIs. Today, though, we are talking about variants. So that same level of care, looking for any of those mistakes in metadata, actually scouring the manuscripts, checking the data, checking the supplemental materials. Those are things that the COSMIC team does in order to tackle creating this comprehensive library of variants. And they are pulling this together from a number of sources, whether it's manuscripts or structured data. So today, focusing on COSMIC, again, this is really high quality data that's been curated for at least 20 years. And ultimately, the amount of effort that they've put to streamline and standardize the metadata terms and the file structure makes it possible to search for your disease terms of interest or search for your tissue terms of interest. And Cosmic also has built a number of really useful tools and projects to make that data as accessible as possible. So today, because we are relying on metadata, I wanted to go ahead and mention just briefly some of the metadata types that happen to be covered. So here, this is where I'm showing some of the various sources of information that one can acquire pieces of information related to a given variant. So we will be providing information about the reference, the source of the material. So that would be the manuscript information. There's also going to be demographics data about the patient. So things like the sex, age, ethnicity, any kind of environmental variables. Those are things that are important to use standard harmonized vocabulary to make sure that across studies and across the entire database, you're able to create custom cohorts and know that you're getting all of the relevant patients or individual samples of interest. For the tumor itself, this is where various details about the histology will need to be recorded. So what's the source of the material? What stage? Are there any other details that might be relevant to a researcher? And then ultimately that classification information, additional details about the sample, all of these things are going to be stored in various tables and cross-referenceable based on a number of standardized identifiers. So I mentioned that Cosmic is continuously being updated. So they have several releases a year, and we've recently come across version 100. So I'm really happy to say that the Cosmic team has put out their new latest release. And with that, there is a ton of more information, and you can always view the specific details at the Sanger website by going to the release notes. So what can you do with Cosmic data? Today, we're going to be talking about using the curated tissue information and other source material info to create a database specific for hepatocellular carcinoma. And then ultimately, the authors would use that to identify potential neoantigens in their HepG2 proteomics. However, there are other things that you can use Cosmic for, and these range from the clinic to the research lab. One particularly creative use that I've come across is potentially using Cosmic to evaluate genomic loci for any kind of mutational impact. So in other words, if the researcher knows or they suspect that there might be some sort of predicted off-target effect from, let's say, a CRISPR mutation or some sort of gene therapy, it's possible to use Cosmic to search comprehensively for any kind of potential mutation that has been detected over the past several years within a window around that genomic location. And then from there is identify and prioritize whether or not those variants that have been detected in the past, are they significant? Do they lead to functional consequences? Are they oncogenic? Another potential use case would be to take advantage of the actionability data that Cosmic has curated. So when it comes to patient reporting or any kind of assembly of basically a clinical report, it's really important to have access to the latest knowledge to know whether or not the variants that were detected have known drug action associated with them. Does a particular variant lead to better clinical outcomes? Does it lead to potential drug resistance requiring alternative treatment? Those are just some of the details that are available in the actionability releases at Cosmic, which again are available through flat file download and implementation with your own clinical reporting pipelines. So with that introduction, I do have just a few questions for the audience. And I also want to open the floor as well to any questions that you might have. But first, one of the things that Cosmic also has is mutational signatures. So these are essentially signatures that are accumulated over time in an individual patient. And the goal would be to isolate which of those mutations acquired over time were from cancer as opposed to just simply from the environmental background or upbringing of a particular person. These mutations signatures can be profiled using tools like Sig Profiler. And we were curious, is that something that you would potentially be interested in using in your own work? I'll go ahead and give about 5 more seconds before I go on to the second question that I want to ask. And then the second question that I would ask would be, how often would the Cosmic team need to update actionability data in order for it to be useful to your research? This is, of course, balancing wanting to have access to the latest cutting edge data versus the amount of time it might take to maintain or update your own internal pipelines. So if you could let us know, that would be fantastic. I'll leave this question up for another 10 seconds or so. So speaking of questions, this is where I, again, want to open up the floor to any questions that the audience might have. So what do we have for that?
All right. Yes, let's look. Okay. First question. Are there any differences between commercial and non-commercial cosmic data downloads?
That's a really good question. The answer is there shouldn't be, but you can always double check on the Cosmic website. One of the things that's really nice about the latest releases is Cosmic has recently updated to a much easier to use file format. So all of those details, the documentation is available on the Cosmic webpage, and the README files are provided in the download files, whether you're downloading from Cosmic or from QIAGEN.
All right. And next question. How does Cosmic choose papers for curation? What types of data do they extract?
Those are definitely good questions. So ultimately, there's going to be a ton of articles that Cosmic needs to sort through. So the Cosmic team is going to scour PubMed weekly for any kind of new articles. And importantly, they're evaluating them manually for inclusion. Ultimately, the Cosmic scientists are going to go ahead and curate things like the variant data, the tumor, histology, sample details, patient information, and any other data points that they can really get their hands on that fit within the controlled hierarchy of information. They do have a full classification system that they publish on their website. So the details for that, if you happen to go to Cosmic's webpage, it would be at the classification. So here, I'll just go ahead and show that URL on screen and just show that resource very briefly.
Are you showing it, Kyle?
It should be, yes.
We are not seeing your screen.
Give me one moment. There we go.
Oh.
Glad we checked that now. So while I have this up on screen in case anyone wants to copy down any of that information, are there any other questions?
Yeah, let's take just one more in the interest of time. And this one is, how do you pick the actionability scores?
That's a great question. That's one I'm actually going to pass on to the Cosmic team to answer.
Fab. Thanks, Kyle. Can you hear me okay?
We can hear you great.
Fab. I was just going to add to your last question about how we decide what we curate as well. So we do tend to have focuses. So for version 100, the Cosmic team decided to go back to our original four genes. So we looked at BRAF, KRAS, NRAS, and HRAS. And then for the up and coming release, we might focus on a specific disease area or a subtype of cancers as well. So there is kind of a thought process that goes behind what we should focus on and perhaps think about what data might be lacking from our data set and how we can enhance it further as well. So back to actionability, the question was, how do we decide the rankings? Was that correct?
Yes.
Yes.
Yeah, fab. So we don't decide them as such. We let the data dictate what we set the actionability ranking as. So I won't go through all the specific rankings because that would take quite a while. But for actionability rank of 1, which is the most significant, there would have to be data to suggest to show that a biomarker, so that could be a specific point mutation or a fusion, is used as a criteria for entry in a drug label that is FDA approved. So yeah, there's different parameters and it's all evidence-based. So we don't just look at specific drugs, we look at the data that's there and it goes into set categories based on the evidence. Yeah, I hope that answers things quite comprehensively.
Definitely. And likewise, for the audience, if there are any further questions or any additional details that you'd like us to clarify, please go ahead and use the Q&A feature. You can find that link just on the sidebar to your left of your screen. So with that, this is where I'm going to go ahead and move into the particular use case. This is where we're looking at this manuscript and seeing just exactly how the authors took advantage of Cosmic in order to generate their HCC-specific library. So here on screen, this is an article that came out earlier this year that's looking at Cosmic-based mutations and using those to enhance the efficiency of identification of any kind of peptides within the HLA-1 immunopeptidome. So if the authors are really interested in digging into immunopeptides, it's really important to be able to identify what they are and whether or not they happen to be cancer specific. And this can become particularly tricky because with variants, you're changing the mapping, and then you need to identify whether or not that mapping is cancer specific, or is that just simply background noise due to the variability present within just the human population. So in order to build that specific library, the typical approach would be to take advantage of some sort of whole exome sequencing approach. So you would take conventional whole exome sequencing data. You would go ahead and use that to derive the protein space, and you could use that to positively identify potential cancer variants. This approach, unfortunately, is not great at detecting things that are rare or low frequency. And if there is a particular cancer type that has a low tumor mutational burden, or TMB, then that can be especially difficult. So in order to supplement any kind of whole exome sequencing data that one might acquire or generate, this group decided that they wanted to go ahead and use the COSMIC database to download hepatocellular carcinoma specific variants. And ultimately, they used this downloaded information as well as a few tricks in order to make it easier to manage, to build a reference library that they ultimately used for their proteogenomic and bioinformatic analyses. So what did they do? Here on screen, I'm just showing essentially an overview of their workflow. They started by looking at all of the cosmic mutations that happen to be present within liver samples that had hepatocellular carcinoma. From there, they went ahead and filtered those to make sure that they were only looking at unique mutations and variants. They were able to use that to generate the specific protein sequences. But from there, the really powerful thing that they did was they actually decided to only take a very small window around each variant. So rather than including the entire protein sequence for their approach, which would result in a rather large data file, instead, they're only focusing just on a very limited window around each known variant. In this case, I believe they used 50 amino acids, but I can go ahead and show the method just to show and verify. So in order to do that, the first step, though, is they need to actually find and download hepatocellular carcinoma specific variants. So how would they do that? So fortunately, the COSMIC webpage makes this surprisingly easy to generate. So using the cancer browser feature on the COSMIC website, this is where it's possible to go ahead and filter any of the available samples based on their annotated tissue, any kind of subtissue details, their histology, and then finally, the specific subhistology and cancer type. Once they have that information, this will generate and provide a list of samples that pass this criteria. And then from there, the authors were able to use this list of 3,000 or so individual samples, and they would be able to then access the downloadable files on the COSMIC website or through QIAGEN. So there are a few different ways that you can go about downloading the genomic screen data that COSMIC has. If you happen to be a non-commercial user, you'd probably be using the Sanger website. So in order to access that, you would essentially go to the COSMIC webpage. You would look at the genomic screen mutants. And then from there, you could choose your reference genome of choice. You could either download the entire library of information, which in this case is only 3.6 gigabytes, or one of the other features that COSMIC makes available is this filtered download tool. And from this is where you could potentially paste in your sample list. And once it's done processing, it would be potentially possible to then download just those samples. Of course, the same information is available at the COSMIC website, at the COSMIC download portion of the QIAGEN webpage. And so this is something that I'll go ahead and just demonstrate how to do these things on screen now. So it looks like the page is not sharing. Let me try that one more time. There we go. So if you happen to be on the Cosmic webpage, this is where you would be able to go to some of the various tools. And one of those tools that is available is the cancer browser. This particular section of the webpage makes it really easy to look for and focus just on the specific details of interest. So for looking at hepatocellular carcinoma, the authors would be interested in liver samples. Once those are selected, in this case, liver doesn't have subcategorization, so we can just include all of the samples. Then we would want to choose whether or not we want to include all samples or if we want to focus only on those that have been positively identified as having cancer. And then finally, we can decide which specific cancer types we would want to focus on. So here, I've focused on hepatocellular carcinoma. This has identified 3,340 samples of interest. So again, to get here, you would go to Tools, and then Cancer Browser. And this would give us access to this particular set where we could filter for those categories. We could also choose which assay type we wanted to focus on. When I hit Go, this is going to load a summary of those particular cancer samples. So in particular, we can see what are, let's say, the top 20 mutated genes within this particular tissue. So we could focus on genes that have mutations, genes that don't have mutations. You can see that we have other information as well that is currently loading. So in this case, for example, this mutation matrix will show the correlation between the top 20 genes in samples. So which mutations seem to be carried together. You can see that we also have a distribution essentially summarizing what the variant types are to see if they happen to have any kind of particular skew. And then near the bottom is where we can actually get our sample list. So here it happens to be showing just 10 entries at a time. However, this is where you would be able to potentially export the information as a comma-separated or tab-separated file. And so by clicking on one of these links, this is going to ask you to log in. And then once you happen to log in, it'll then ultimately download that particular file. Then if you wanted to access the Cosmic data itself, this is where you would want to go to the data section. This is where you can access some of the collections that are really incredibly useful, such as the actionability information or any kind of mutational signatures. For overall data download, you would just click on this first link, though. And this will bring you to this page where you can very easily download these sample files if you wanted to get an understanding of the file structure. You would also have access to this changelog, which can give particularly useful information about how the files are organized. So within this changelog, for example, there happens to be a rather useful chart which will show what some of the various file names and headers are that could be potentially used to, say, sort through the individual items. So essentially, what fields could be used. So here, I'm showing how one might connect some of the files together. And this would be using something like the cosmic phenotype ID or the gene ID, sample ID, fusion ID, paper identification, etc. But going back to the general information, here I'm showing the genome screen mutants. This is the section that the authors were interested in using, because this is all of the coding point mutations from any kind of whole exome sequencing or other genome-wide screens from the current version 100 release. Importantly, all of these also have clear file descriptions giving details about the headers. So this is where, for example, if you needed to use the cosmic phenotype ID, this would be another way to retrieve the tissue and histology information and have that be standardized across all of your samples. Another useful field way at the end is the somatic mutation status. So this would be the ability to check to see whether or not a particular variant has been confirmed to be somatic because they have it reported. They have the clear-cut comparison between a case versus control. Or if the variant is of an unknown origin, that will be highlighted, which means that we just don't have access to a matched normal tissue from the same individual. So I mentioned that the authors would typically download the full file, but it is also possible to download only a slice. And the way to do that would be to use the filtered downloads. So here, unfortunately, to use this feature, I need to go ahead and authenticate. Give me one moment. And under this filter, this is where if you only wanted to focus on specific gene symbols, specific primary sites, or sample names, it would be possible to input this information. If you happen to be a commercial user, you likely would be downloading this instead from ideally an internal resource at your company. But if you're responsible for managing the downloads, you would go to the QIAGEN Digital Insights webpage, and you should have access to, after going to the product login page, the various cosmic resources and other data that you might subscribe to. And once you authenticate here, this is where you would be able to access any of the goodies that you subscribe to or license. And this is where you can find all of the cosmic download files, as well as additional tools such as Anavar. This cosmic download page, the main heads up that I would give is that you do need to use this drop-down menu in order to choose the correct release. So here, if I want to choose Cosmic version 100, I would go ahead and go to the drop-down menu, choose Cosmic version 100, and then this is where you would be able to go ahead and scroll down and identify and download the genome screen mutant files. So again, just to summarize, you would have essentially two options to download the data, depending on whether or not you're a commercial or an academic user. But in either case, you would be able to download the complete data, and then from there, you would be able to go ahead and very easily take advantage of the various sample names, or you could use the classification details in order to focus on the specific phenotype of interest. So with that, again, I'm going to pause for questions. And just to keep things fair, I'm going to go ahead and ask you, the audience, one question. So given that this particular application relies on the curated metadata in COSMIC, we were curious if you use curated metadata for your own research needs. So this could be because you're looking to find any kind of specific cell line or tissue or tumor type of interest. You might want to be assessing market size by taking advantage of demographic information or mutation frequency details. You might need this metadata when reporting clinical findings to medical providers or patients. If you're using it for a really creative purpose, you should let us know, because we would be really happy to discuss that. I'll leave this poll up for another 5 or so questions, and then open the floor up to your questions.
All right, next Q&A. So let's see. In the interest of time, let's just do maybe 2 this session, and then we'll save the rest for the very end. Okay, so first question is, let's see. How are the contradictory consequences for specific mutations published in literature handled in cosmic curation?
That's a great question. And that is one that I'm going to pass off to the Cosmic team.
Yeah, thanks, Kyle. Yeah, I could take that. So if there's any contradictory evidence, it would all get incorporated into the COSMIC database. We aren't subjective, we do quality control, but we don't look at contradictory evidence. Yep, all somatic mutations that meet our QC steps would get integrated into our database. And there's quite a vigorous process that's involved before data gets incorporated. Anything you would add to that, Jenny?
No, I don't think so. Rebecca, I think you've done well. I'm not sure I really understand the concept of contradictory information. I mean, there are mutations that are present in a particular patient, another mutation is present in another. So we record the mutation. The consequence of the mutation, would be whatever the publication has put forward. So the amino acid change or that sort of thing. I'm not sure I understand the contradictory consequence that may arise.
I would say that we actually encounter this a lot in ingenuity pathway analysis. We also curate any conflicting information because there might be experimental details that would lead to a different observation from different authors. Or likewise, it could just be a good old-fashioned author fight. But either way, we want to be sure that as much quality information is put into your hands, and we would rather display all of the available knowledge rather than inappropriately filter out.
So I think, as Rebecca says, there are very rigorous controls in place. So, the curators are going through everything, but our job is essentially to record what is in the publication. So as long as that publication or the information passes a kind of, check for quality, that's going to be included in our database.
Perfect.
All right.
And-
And oh, sorry,
and-
Kyle.
I was going to say, any other questions?
Yes, yeah. We have one more for this break. Why does it matter whether they are HLA type 1 peptidome or HLA type 2 peptidome? Can they both be relevant therapeutically for vaccine generation?
So that is another good question, which again, I'm going to pass off because unfortunately, I am not an immunologist. I just love messing with the data.
Yeah, I would agree there, Cal. I think it depends on the scientific question you're trying to address. And within Cosmic, our expertise wouldn't be within vaccine generation either.
So in this case, I would say that fundamentally, the main thing would be to make sure that whatever approach you're using, that you have good variant information to cross-compare it to. Because no matter what you're detecting, if you need to see if it's cancer specific, you want to compare it against a database.
All right. We will save the rest for the end if that works for you.
Alrighty, thank you. So with that, I do want to go ahead and briefly cover details about what the authors basically came across and information that they reported. So in this particular case, because they were able to access quite a bit more information using Cosmic as opposed to using their downloaded whole exome sequencing data, this really increased the number of mutation sites as well as the mutated genes that they detected. And importantly from that, when they were actually using that information to identify HLA-1 immunopeptides, they still had extremely good overlap between both items. So in this case, they were able to, using both methods, generate this high number of potentially useful mutant peptides. And from that, this also led them to basically reason that it may be in fact possible to use a database-only approach without any kind of experimental data when tackling this type of problem. And from the data, in this case, one of the things that they went ahead and provided was a list of the potentially interesting and relevant mutant peptides. And of course, from there, they would need to go ahead and then try to predict which ones of those would be antigens, which ones would potentially be binding to HLA-1 molecules. And for that, they went ahead and took advantage of some other tools and resources. And they found that they identified about 5 of these immunopeptides from the experimental data, which was up from 1 that they were able to identify through the whole exome sequencing screen. One of the things, though, that the authors did caution is that when looking at these results, they did notice that the predicted peptides from Cosmic did sometimes show lower potential binding. So they wanted to be sure to highlight that it is important to always follow up and verify and validate the experimental results, just to make sure that you're removing any kind of peptides with poor spectral quality. You're removing anything that doesn't necessarily have a good affinity binding. So with that, I'm going to go ahead and just quickly summarize what we discussed today. I went ahead and gave you just an introduction to some of the various manually curated data that we have available here at QIAGEN, as well as from Cosmic. With that, there's a number of different methods available to download and integrate that data directly into your analysis pipelines. So you're not dependent on using the Cosmic webpage. you are able to download the VCF files, or you can download the tab separated or comma separated data. And with that, you would be able to then implement any of the details that you would need for any potential application. Ultimately, this is going to be dependent on all of the metadata that has been curated. So this metadata, again, is something that every cosmic scientist is looking for whenever they're reading a manuscript. And once they fill this information out, all of the terms are going to be harmonized and using controlled vocabulary, which will then enable applications such as searching for just very specific hepatocellular carcinoma samples, and then downloading those resulting peptide sequences or protein sequences in order to generate a reference library for alignment of mutants. So with that, I want to go ahead and pause one final time for questions just to see if there are any last minute questions that we or the Cosmic team can address.
Yes, absolutely. And just as a reminder, we will follow up on any questions that aren't answered here via email. So if you do still have any questions, go ahead and submit them. Even if we don't get to them, you will get a response. So let's go ahead and jump in though. Is there any plan to classify the tiers according to standard guidelines such as AMP?
So that's something that I know that we do with the variant information that we use for QIAGEN Clinical Insights. We have a really useful tool to parse through your VCF files for clinical reporting. However, I'm curious from the Cosmic team about that specific question.
Thanks, Kyle. Yeah, it's something we do get asked a lot with our actionability data. The one difference between ourselves and the AMP ASCO guidelines is the fact that we don't have subjective opinions or have a panel of clinical experts, which is what AMP ASCO guidelines are based on, particularly Tier 1 Level B. So there is a lot of questions we do get around that. And a lot of people do ask whether we could map to them. The answer is we can't map directly because we don't have that same expert panel. But it is something we are looking into about how we can maybe map, but clearly state the differences between what we do in actionability and what is different within AMP ASCO guidelines as well.
Awesome. So essentially, if you want to implement that into your own research, again, those flat file downloads, access to the raw data should make it easy for you to at least map any kind of external source that you would have. Or if you want something that is turnkey and ready to go, QIAGEN Clinical Insights Interpret would have access to all of the various AMP guidelines for variants and reporting.
All right, thank you. And just our final question here, since we are running a little bit over on time, are there tools for uploading a VCF file and getting automated variant prioritization and annotation as output?
Yeah, so that QCI Interpret tool provides a lot of robust filtering and reporting for clinical or translational research lab samples. And that is going to take advantage of all of QIAGEN's curated findings, of which Cosmic forms a key part. However, I think that a better potential answer for many researchers would be to take advantage of Anavar. So Anavar provides a Perl script approach to do it yourself. And in this case, this framework that they provide interacts directly with Cosmic's flat file format. So that can be a way to easily, well, more easily get started, especially in your research lab.
All right. Thank you for your responses. This does conclude our session. Thank you again, Drs. Nilson, Wilding, and Steele for your time today. Thank you as well to the audience for joining and for your active participation. We really appreciate it, and we hope it was mutually beneficial. Again, any questions we didn't answer today or any asked after the conclusion of the presentation during the on-demand period will be addressed via the email you provided at registration. We've also put both QIAGEN and Cosmic's contact info up on the screen for you here for anyone who might want to reach out later with additional questions or, again, interesting use cases. Again, LabRoots will notify you via email when the recording of this presentation becomes available for replay. We highly encourage you to share that email with your colleagues or anyone who may have missed the event and might find it interesting or useful. Until next time, stay safe and be well, everyone. Goodbye.
Take care, everyone.